So the next type of spectroscopy we can look at is infrared spectroscopy. So this relies on the idea that all covalent bonds, or all the organic species, have bonds in them that absorb particular energies. So let's say you've got ethanol. It looks like this. You take that sample and you shine a beam with a range of different energies in it, so ranging from 200 to, I think it's like 4,000. Yeah, 4,000 centimeters to -1. So you shine that variation of energies at this molecule, and what will happen is the bonds within the molecule will absorb some of the energies in that particular packet. So a carbon to hydrogen bond absorbs energy between 2,800 to 3,100 centimeters to the -1. So when the light actually comes out through the other end, so when it comes out through the other end now, that packet of energy there will be missing. You'll have this oxygen to hydrogen bond, which will absorb energy, let me just check, 3,200 to 3,550 centimeters to the -1. So that little packet will be missing. This will absorb some energy as well, and this will absorb some energy as well. So if we can imagine a little graph, you've got wave numbers, which is equivalent to energy measured in centimeters to the -1. So it's done backwards, don't worry about why it goes backwards for now. So 1,000, 2,000, 3,000. So that's the packet of energy that we throw at the molecule initially, before we've passed it through, in terms of intensity, so, or transmittance is the term. So in terms of transmittance from 100 down to 0, before we put the sample there, actually all those different energies are going through, so nothing's getting in the way, nothing's absorbing those energies. As soon as we put that molecule there, what happens is these bonds start absorbing some of those energies, and we start getting, so the little peak, so between 3,200 and 3,550 we get a peak, where actually the amount of that particular gap of energy that's been transmitted has dropped, so there's less of that type of energy getting through. Same for this little range here, so we'll have a little peak between 2,800 and 3,100, and we'll have another peak for some of the other molecules. So to speed things up, I'll just show you some examples, and it'll save me having to make it again. So here's an alcohol. I think that's clear enough. I think I say that's clear enough. So we've got propan-1-ol in the diagram. This is the infrared spectrum of propan-1-ol. So you can see here we've got a carbon to oxygen peak. If you look in the data booklet, you'll be able to see a carbon to oxygen peak should appear between 1,000 and 1,300, which that one does. We've got a carbon to hydrogen peak here, which is between that 2,800 and 3,100, and then an OH peak here. If we go further down, so this is a ketone. We've got, what was that, pentan-2-one. So it's got a carbon to oxygen double bond. Carbon to oxygen double bond should be between 1,650 and 1,750, so it's that one. And then we've also got carbon to hydrogen peaks, which are over here. Carboxylic acid tends to look like that. So the main peaks to look out for are the OH and the carbon to oxygen double bond peaks. So there's the CO double bond peak, and there's the OH peak. You don't really tend to be that concerned with carbon to hydrogen peaks, because you're trying to get information about the molecule, and because it's an organic molecule, you can see all of these have got a CH in them, so actually that peak isn't that useful usually in helping us determine anything about the molecule. So there's carboxylic acid, and I think that's it. One thing to highlight to you before we try a question is the fact that if you look at an alcohol, so alcohol's got an OH peak, it's quite narrow, so you can still see the CH peak, whereas if we go down to a carboxylic acid, it's still got an OH peak, but the OH peak is really broad, so it kind of like muffles the CH peak, and you can't really tell that there's two separate peaks there anymore. So those are the different types of infrared spectrums. Let's have a go at a question right then. So here's an example of a really basic question on it. You've got propan-1-ol has this infrared spectrum, and it says, using the datasheet, state how the spectrum confirms which functional group is present in propan-1-ol. So they're interested in just the functional group that's present. If you look at the data booklet and you look at what this peak here corresponds to, that's corresponding to a carbon to hydrogen bond. We're not interested in that because it's not a functional group, so that's not going to get us any marks, but that is a carbon to hydrogen bond. This peak might be a carbon to oxygen bond causing it, but the peak that confirms the functional group is this one here. So that is a oxygen to hydrogen bond. So you'd want to say in this question, peak between 3,200 and 3,500 indicates the presence of an OH bond and therefore an OH functional group. So that's one particular question. I think I found another one later on. So if you look at this one now, so they just talked about propan-1-ol. If we look at this, I don't even have to read the question, you know that's a carboxylic acid because that peak is really, really broad. So there's a carboxylic acid present in there because it's muffling that CH bond. They do tell us it's ethanoic acid, but one day they might not, so you need to be able to recognise that as well. So that pretty much ends infrared spectroscopy, the theory. What they tend to do with infrared spectroscopy is they tend to integrate it into, so if I find a bigger question now, they'll tend to integrate it in with other types of spectroscopy. So you can see here there's it's part B, it's got a mass spectrum in it, and it's also got an infrared spectrum, and you need to get as much information out of both of those in order to work out the answer. We'll practice one of those in a later video. But that's all for now.